tonight is I've sort of divided the talk into two sections. And let me just start out by saying that I invite you to ask questions throughout. I don't have a strict agenda for the evening, and so if there are things that you're curious about or seem unclear or really whatever strikes your fancy, please, I love to be interrupted. That makes for a fun evening. So what I thought I'd start out on is really some background material, or I should say general material on sort of what are these things we call telomeres, what is the enzyme that is really the main focus of my laboratory's research efforts called telomerase, and how does it relate to human health or when human health goes awry in disease states. And so the first part of the conversation will really be sort of general telomere and telomerase biology in human disease, as the title indicates, and then toward the end I'll get a little bit more technical and tell you a little bit about what we specifically are doing to make contributions in this area, the exciting area of science. And so I'd like to start out, so there's a lot of popular culture, knowledge, and awareness about telomeres. I've learned over the years since I've been studying this topic for a little over a decade now. So just by show of hands, I'm curious how many of you already know what a telomere is? Yeah, so most of you, right? And so most of you probably have read or are aware that telomeres are these interesting structures that cap the ends of our DNA genomes, or our chromosomes that make up our DNA genomes, and I think a very appropriate metaphor, which I really can't take personal credit for, because the first time I heard it was from the Nobel laureate Elizabeth Blackburn,
we don't have that APC anymore. I think that's the rotating
the molecular
one.
basis of telomeres and ultimately the enzyme telomerase together with her student Pal Brother.
See one right now, yeah.
So she compares telomeres to this small piece of plastic at the end. Oops. So in my religion, that's good luck. So that's good. When you break the glass. So that's good. That's a good sign. So this small piece of plastic on the end of a shoelace, which is called an aggregate, right? Its function is to protect or safeguard the genetic material, the material that comprises our genome, and when that small protective cap is compromised, the structure is somehow damaged, then you end up with a tattered shoelace, and so that's where this metaphor sort of arises. And so let me now tell you in a little bit more molecular detail what is a telomere. Okay. And so here's a nice picture. This is what is called a metaphase spread. It's basically all of the genetic material from a single nucleus of one cell spread at a particular time of the cell cycle when the DNA is nicely condensed and you can get these pretty, what are called metaphase chromosomes. So in this sort of fluorescence microscope image, everything you see in blue here is the DNA, most of which is interestingly not necessarily encoding specific genes, but has other interesting functions. And then what you can see, the way that this image was prepared, there's a special stain that specifically binds to and lights up the telomere structure. And that's what you're seeing as these little yellow dots at the end of each of the chromosomes. Okay? And so indeed, this idea of the plastic cap at the tip of a shoelace is very apropos. Okay? I mean, it's a fair comparison. So if we now take this into a more sort of schematic view, so now I'm just drawing that same sort of picture in a cartoon, I can tell you at first that the DNA that we find at the ends of our linear genomes, or the chromosomes, is not just sort of your average piece of DNA. Okay? It has very specific sequence. So some of you may know that DNA is made up of 4 basic building blocks that often go by the letters A, G, C, and T, right? And what you'll notice here in this drawing that I've illustrated is that the sequence at the end of our genomes that really make up the foundation, the sort of lowest level of telomere structure, have a very specific sequence, and it's very repetitive. And so in you and I, so all of us sitting in this room, right, have 6 base repeats, TTA, GGG, TTA, GGG. But this is not drawn to scale, but basically at the ends of our chromosomes, you have thousands of these small 6-base repeats. You can think of them as little words, right? That mark, I'm a telomere, okay? And then what's interesting about that is that, as is often the case, specific DNA sequences serve a role to actually recruit specific proteins in the cell. In the case of telomerase and telomerase-associated proteins, don't worry about all this fancy nomenclature. There's a particular protein complex which is often referred to as the sheltering complex. And it's called the sheltering complex because it shelters or protects the ends of our genomes, right? So why might we need to protect the end of the genome? That's a fair question to ask. Why is that an important function for the telomere? Well, the answer is that every day we go out, particularly on days like today in the sunshine. Some of us, eat junk food from time to time, right? We put things into our body, or we're exposed to radiation, and that causes damage in our DNA, right? And sometimes that damage can lead actually to full breaks in DNA, what we would call a double-strand DNA break, which is basically like taking a piece of string and cutting it with a piece of scissors, right? Or a pair of scissors, I should say. And that sounds like a terrible situation, but fortunately for us, we have very robust repair machinery inside of our cells that are constantly on the hunt, on the prowl, for these sites of DNA damage, looking for them, detecting them, and repairing them. Thank goodness, right? So all this damage all throughout the day, it's happening all the time, and it's being repaired, hopefully, all of the time. And when things go awry with that repair process, that itself can lead to disease states in organisms like you and I, right? And so when, through the course of evolution, multicellular organisms began to evolve genomes that consisted of linear long pieces of DNA that can be compared to like a piece of string. E. coli, the bacteria that we're all familiar with, right? That lives in our guts, right? It has a DNA genome too, but its genome is a circular piece of DNA. So it doesn't have ends on the DNA, right? And so we have a unique challenge, or our genomes have this unique challenge of having a bunch of ends, right, that are perfectly natural, right, and are in fact supposed to be there, and should not be detected by DNA damage repair machinery. In the event that they are detected by repair machinery, one chromosome can fuse to another, and then that, you can imagine, gives rise to problems. Let's say a cell needs to divide, and the chromosomes can't properly separate or segregate, is the term, right? So this sheltering complex, one of its major functions, very often when I'm talking to students, or town folk, as the case may be, right? The question is, well, why do we have telomeres? And if I had to just say in one simple statement, it is to differentiate the natural ends of our linear genomes from sites of random DNA damage that would otherwise be repaired, right? And so these proteins bind, right, and sort of tuck away or hide or shelter, right, the ends of our chromosomes. And really that is what makes up the essence of a telomere. It's proteins and DNA protecting the ends of our genomes from this very robust repair machine. Okay? So that's a little bit of an introduction to telomeres. Okay? So that's the good news. Okay? Perhaps the bad news is that each time a cell divides, so we're all familiar perhaps with the idea that cells, many of the cells in our body need to divide as we develop new tissues or respond. Say if you're a cell that's part of the immune system and you're responding to some foreign molecule, there's a lot of cell division that occurs. And it was recognized very early on that, in fact, it dates back quite a long time. So the structure of DNA, which was first proposed by James Watson and Francis Crick, based on work from people like Rosalind Franklin, in the early 1950s, so it was 1953, April 1953, when the DNA structure was first proposed, the double helical structure of DNA. And James Watson realized, and many others, but James Watson wrote in an important paper a couple years later, that they recognized that there was a problem, the so-called end replication problem. that was sort of derived from the very nature of the structure of the DNA itself. And the essence of the end replication problem is simply that, without going into too much detail, is that the machinery that copies our genetic material every time a cell needs to divide has a hard time completing the task, okay? So when it goes all the way to the end of the linear genome, it can't finish the job, okay? And that was recognized a long time ago, right? That there might be a problem, the so-called end replication problem. And in fact, many experiments from that time to present have proven that this is in fact the case, that the ends of our chromosomes gradually shorten as a result of this so-called end replication problem. Now, in a few moments, I'm going to argue to you that calling this a problem is a bit of a misnomer, okay? And so just tuck that in the back of your mind, because I would argue that this end replication problem is actually a very natural component of cellular function, which I'll describe to you in a moment. But those of you who raised your hands and said, yes, I know what a telomere is, probably also know that as we age as organisms, right, or our cells age, which for me, it's easier to understand and think about. I have a hard enough time understanding how a cell works, let alone an entire organism like you and I. Our telomeres in most of our cells get shorter. Okay, so that's what this slide is showing, that if you were to somehow measure the length of the telomere as one cell turns to 4, turns to 2, 4, and 8, the telomeres get progressively shorter and shorter, okay? And something that had been observed was that when telomeres get to sort of a critically short length, a cell ceases to divide, okay? It stops growing, okay? So that's like, you might think that's a problem, right? There are certain cells in our body that need to divide many, many times. We would say these cells have a high level of sort of proliferative potential. They're going to divide and grow. An example would be, for example, a stem cell, something that many of you I'm sure are familiar with. A stem cell, a progenitor stem cell, hasn't quite decided what kind of cell it is going to be yet, right? Sort of like a young child that hasn't decided what it wants to be yet, right? And so it needs to divide many times during a process called differentiation. And as such, in those particular contexts, this sort of end replication issue is in fact a problem, right? Because if you hit this sort of wall of cell growth before you've been able to differentiate or turn into, say, a skin cell or an epithelial cell or a bone cell or a component of the blood, then that would be problematic. And so most cells that are rapidly dividing, like stem cells, express this enzyme telomerase to sort of combat or fight against this telomere shortening process that I just described. Yeah, question?
I'm wondering, does the shortening process accelerate with age, or is it a steady thing that starts upon birth?
Right. So the question is, does the telomere shortening accelerate as you age, or is it sort of a linear decrease over time? And I would say, so here's one of my major caveats of tonight. So if you came here tonight thinking I was going to tell you what the fountain of youth is, for example, you're going to be disappointed, okay? I'm afraid. And so at the cellular level, to the best of our knowledge, it's a fairly regular shortening rate, okay? At the sort of more complex level of the organism, it may be that in certain tissue subtypes, right, and in certain environmental contexts, the rate is faster than others. That's certainly feasible. But I think it's sort of the paradigm is that really the shortening is sort of directly proportional to the number of times a cell has divided, okay? Which is sort of saying it's a steady progression or regression, okay? So one of the early links between sort of the enzyme telomerase, this connection between telomere shortening and the ability of a cell to continue to divide, was made in the early 1990s by a group down at MD Anderson in Texas. And what they were doing, so in the mid-80s, Liz Blackburn and Carol Goddard discovered telomerase. Everyone was very excited about that. It provided a very elegant solution to the so-called end replication problem. But then following on that, researchers rapidly recognized that there was a very strong association between telomerase expression and cancer cells, human cancer cells. Okay, and so at that time, I think in that initial seminal study, they studied something like 200 or so solid tumor subtypes, and they asked the simple question of, is there telomerase present that I can detect or not, right? And what they found is that about 9 out of 10 cancers, and that number stays true today. So that was in the early 90s, and so this is a very robust result. So it turns out that one of the ways that cancer cells sort of bypass this challenge is by turning on this enzyme, okay? And everybody got very excited at that time for obvious and appropriate reasons. In developing strategies, I'm sorry, question.
Is the whole telomere made out of calcium proteins, or is there other components?
Sure. So the question is the whole telomere comprised of DNA and only the sheltering proteins that I alluded to, or are there other factors present? And there are definitely a lot of other factors present. And in the interest of simplicity, I'm not talking about them. Okay. But yeah, it's actually a very interesting question because research relatively recently has shown, for a long time people thought, in fact, that telomeres are these specialized structures that have completely different protein composition than the rest of the genome, right? And now what we're learning is that it's a much more nuanced distinction between a telomere and the rest of the genome, which is sort of a talk unto itself. Okay, that's a good question. And so that raised a lot of interest and motivated a lot of research to this day at sort of understanding more about this curious enzyme telomerase. And armed with that knowledge, might we be able to better diagnose and even better treat cancers in more effective and targeted ways. Okay? And to this day, that's what motivates my own laboratory's research. And there are, I would say, thousands of labs around the world who are studying this exciting topic. Okay? So to sort of put into a context the role that telomere shortening plays in sort of regulating cell division, it's useful to think of this concept of the cell cycle. Okay? And so a cell doesn't divide willy-nilly. Right? There's a lot of regulation that goes into turning one cell into two. Okay? It's a very elaborate process that can be subdivided into 4 general stages. There's a growth phase, and then a phase when the DNA is replicating, and then a secondary growth phase, so-called G2, and then this thing called mitosis, which is when now all of your genetic material has hopefully been appropriately duplicated, and now it's going to segregate into daughter cells, and then eventually a process called cytokinesis occurs, which is actually the cell division, sort of the invagination of the membrane that it encloses the cell to sort of bud off into 2 cells, right? And so what I wanted to point out here is that you'll notice that these red bars, right, this bar here, here, and here, these are so-called checkpoints. So these are sort of fail-safe measures that the cell has evolved to make sure that, for example, I'm ready to go from this initial growth phase into so-called S phase where the DNA is going to be replicated. So to put that into more sort of simple terms, this initial growth phase, a lot of what's happening is you're ramping up machinery that's required to replicate your DNA, right? And you don't actually want to get into that process or that business of replicating DNA until you're poised and ready, right? And so there are specific proteins that sort of halt the cell at these different checkpoints and regulate progression round and round through the cell cycle. And in the context of cancer, which many of us sort of think of and appropriately so as uncontrolled cell growth, right? Where the cell is just going around this cycle.